In today's class of the molecular basis of inheritance, we are going to take up a topic, regulation of gene expression. In my previous video, I have told you regarding the regulation of gene expression in case of the eukaryotic cell is that it takes place at various levels, isn't it? I told you that it takes place at the level when the replication and transcription takes place in the nucleus of a cell. Then after that, when the splicing takes place and the introns and the exons are separated, the introns are eliminated out and the exons are again joined together, and then there is a capping and a tailing, and then finally the ready mRNA comes to the cell cytoplasm where the translation takes place. Now the translation also takes place in case of eukaryotic cell in a very regulated manner, in which we have seen that in case of the translation process also it is because of a specified that is your tRNA containing the specified amino acid and coming and binding with that of the codon present in the mRNA, and then with the termination codon there is after the termination codon there is a flank which is having some base pairs for which there is no that is formation of a gene, or before the that is translation also there is a flank which is not at all being translated into any protein. So regulation is at the translational level also. As far as the prokaryotic cell is concerned, mainly in case of prokaryotic cell, the regulation is at a transcriptional level. You see, suppose if we take an example of E. coli bacteria. Now this E. coli bacteria needs substrate for getting energy. Say for example, glucose is a source of energy. But suppose if the E. coli bacteria is allowed in an environment where there is lactose instead of glucose. Now what will happen, because lactose now is a substrate instead of glucose. Now what will happen is that in case of this new substrate of lactose, now there will be an Operon system, a working system to utilize this lactose or to break down this lactose and make it into ready available form of energy. That is, it will be broken down into glucose and galactose by the help of certain enzymes. And now these enzymes will be decided by the sequence which is present in the DNA of that E. coli cell, which in turn will be at first transcribed into mRNA. Then there will be a lot of structural genes because it requires more than one gene, and there will be only that is the translation of the protein will be with respect to all these three structural genes, and as a result of it the enzymes which will be produced, which is more than one, will be now making the cell possible to utilize the lactose which is in the environment. So in this case you see the environment or the substrate is regulating the mechanism in case of the prokaryotic cell, which is the E. coli cell. So this was actually proposed by that of two scientists, Jacob and Monod, and this is more commonly known as your Lac Operon. The word lac has come from lactose, and Operon means the operating system, that how the lactose will be utilized by the E. coli cell when the substrate is lactose present in the environment. In case of Lac Operon, the polycistronic structural genes is controlled by that of a common promoter and regulatory enzymes. Now suppose this is the segment of the DNA which needs to be transcribed for producing the enzymes which will be acting upon the lactase to decompose it or to break it further into glucose and galactose, and then that will be utilized as a ready source of energy by the E. coli bacteria. Then it will be having a part of the regulatory gene just before the promoter region. Now what is this promoter region? Promoter region is the place in the DNA where the RNA polymerase will come and bind. So this is the position where it will be binding. When it will be binding? When in the medium in which the E. coli is growing is having lactose in it. But suppose if the media is not having lactose in it, in that case what will happen? There is no need to operate the system, there is no need for producing of the enzymes or formation of the mRNA with respect to these three structural genes, because it is not required, because it is not present in the media, lactose. So why will it unnecessarily make make the structural genes, isn't it? So in that case, when there is no lactose in the media, then what will happen is that to prevent the that is RNA polymerase to come and bind with that of the promoter region, there is another very small region next to that of the promoter, which is called the operator region, from where the transcription process will start. Now what happens is that prior to the promoter, there is a regulatory gene which is called I. Now I here does not mean inducer, I here means inhibitor. Now this I, which is the regulatory gene, it will synthesize, or it will be producing an mRNA which is called the repressor mRNA, okay, when the lactose is not in the media. Now as a result of this mRNA, repressor mRNA, it will be producing a repressor protein. Okay, now this repressor protein will now go and bind with the operator region just after the promoter region, from where the transcription of the structural genes will start, preventing the RNA polymerase from transcribing the operon. So RNA polymerase cannot bind in presence of this repressor protein, which is produced by this inhibitor gene, which is a kind of regulatory gene. See, it is getting regulated. So here the regulation of the gene expression is happening by the help of another regulatory gene present just before the promoter region, from where the transcription process will start. Okay. Now this is all happening when there is absence of inducer. What is the inducer? Inducer is a substrate. And what is the substrate? Substrate is a lactose. That is why the name has been given as your Lac Operon. Okay. Now next, suppose now this scenario is changed. Now what is this change in the scenario? That is presence of inducer. That means now the bacteria is allowed to grow in a media which is containing the lactose. Now when there is a lactose has to be utilized, lactose has to be broken down, isn't it? So if the operator region is blocked by the repressor protein, how will the synthesis of these structural genes will take place? And as a result of it, the breakdown of the lactose cannot take place, isn't it? So the repressor protein has to be removed. So how it will be getting removed? Let us see that. Now in this case, when lactose is present, what will happen is that this regulatory gene I, which is the inhibitor, will produce the repressor mRNA. Okay, it will produce the repressor protein. Fine. Now since the inducer is present, what is this inducer? Lactose, or one form of lactose which is called allolactose. Now this inducer will come and bind with this repressor protein. Once it binds with the that is your repressor protein, there is a change in the structural configuration of this repressor protein. You have read it in class 11 that there is some inhibition when there is any inhibitor is coming, or any inducer is coming and fixing or is combining with that of a protein. It changes the conformation of the protein. So once the conformation of the protein is changed, now it cannot come and bind with the operator region next to that of the promoter region, because it is now changed, it has changed its conformation. So there's a lock and key process. So that place where it is going to go and bind, that due to the change in the conformation, it can no longer bind over there. So as a result, now the pathway is clear. Now as a result of clearance of the pathway, now the RNA polymerase can easily bind with that promoter region and start making the that is your Lac mRNA. Okay, lactose mRNA. Now transcription of the lactose mRNA, when it will be done, then after that will be the translation, that is the translation of this Lac mRNA. And what will be the translation's result? There will be 3 enzymes. What are the 3 enzymes produced? The first one, that is with respect to the structural gene Z, it will produce beta galactosidase. As the name suggests, galactosidase, that means it will be now breaking down the galactose into glucose. It will be breaking down the lactose into glucose and galactose. Okay, now once it has been broken down, now it has to be utilized, or else what is the use of lactose in the medium? So the next structural gene, that is a Y, will produce another it will be translated into another enzyme or a protein which is called permease. Now if you remember, Permease is a membrane protein, that is, it is found in the cell membrane, which allows or which increases the permeability. So as a result, the entry of the lactose from the surrounding into the cell can be easily made. And then there is another structural gene, which is A gene, and it will be producing the enzyme trans-acetylase. Now, trans-acetylase as such, the function is not so clear, but it has been seen that trans-acetylase along with the permease helps in the movement of the lactose within the cell, so that the beta galactosidase can break down easily into into your glucose and galactose, and as a result the ready source of energy is available for the bacteria to be utilized. So this is all about your Lac Operon in presence of an in absence of an inducer and in presence of a inducer. When there is no inducer, that means when there is no lactose in the medium, in that case the regulatory gene I will be producing a repressor mRNA. With respect to it, a repressor protein is produced, which will be binding with that of the operator region, preventing the RNA polymerase to transcribe the structural genes. As a result, there is switching off of the Lac Operon, and when there is presence of the inducer, that is lactose is there, the regulatory gene which is producing the repressor mRNA, as a result of it the repressor protein is produced. Because of the inducer or the lactose which will be binding with this repressor protein, it makes the repressor protein inactive. As a result, it gets released, it cannot bind with the operator region, so the region is free. Now easily the RNA polymerase can come and bind with that of the promoter region, and all these structural genes can be transcribed, and then it can be translated into the respective that is enzymes to be utilized. These enzymes will be utilizing the lactose into glucose and galactose, and as a result easily the utilization of lactose can be done by that of the E. coli bacteria. So such type of regulation of an operon by the help of a repressor protein is a kind of negative regulation. Okay, positive regulation is not in the syllabus, so we are not going for that. Negative regulation is done by a repressor protein, and the example is that of a Lac Operon. I hope you understood today's class well. The next topic is about human genome project, which we will be taking in our next class. That's all for today's class.